Hello everyone, I'm Himanshu Saxena from Physical Research Laboratory of India. I'm delighted to be here today. And before I start, I would like to thank the organizers for giving me this opportunity to present my recently published work titled Contribution of Carbon Fixation Toward Carbon Sink in the Ocean Twilight Zone. In this work, we have tried to assess the role of chemoautotrophic carbon fixation towards CO2 drawdown, in addition to the role of carbon fixation occurring in the euphotic zone. I would like to start with a brief of carbon and nitrogen cycle in the marine environment and the motivation. This will help us to realize the motivation behind conducting this research work. Phytoplankton are the marine functional unit of food web. They are the primary producers that assimilate dissolved carbon dioxide gas to their organic matter. In addition, they also assimilate nitrogenous nutrients, which could be in the form of ammonium, nitrate, nitrite, or dissolved organic nitrogen. Most of the organic matter is produced in the sunlit layer of the ocean, which might range from surface to around 200 million tons. As this organic matter sinks down, As this organic matter sinks down, it gets remineralized with the help of oxygen into the inorganic constituents of the organic matter, such as ammonia or carbon dioxide. This process of remineralization leads to a decrease in oxygen concentration in the subsurface water. This zone is popularly known as oxygen minimum zone. This oxygen minimum zone provides a suitable niche for a range of organisms and the associated mechanisms. For example, the ammonium is converted to nitrate and nitrite and ultimately nitrate through the help of nitrifying organisms. But this reaction is not restricted to the oxygen minimum zone. It could happen in the oxycline region as well. Another reaction is Anammox reaction, which is performed by Anammox bacteria. These bacteria, in the absence of oxygen, utilizes other oxygenated nutrients such as nitrite, and along with the help of ammonia, it converts the nitrogenous nutrient into dinitrogen gas. Additionally, similarly, the denitrifiers reduces the nitrate through a chain of reactions to nitrogen gas in the absence of oxygen. Notably, these nitrification and Anammox organisms are chemoautotrophic, and some of the denitrifiers are also autotrophic, meaning in addition of remineralization through which they produce CO2, they're also able to assimilate the dissolved carbon dioxide gas to their organic matter, meaning they are also playing a crucial role towards CO2 drawdown. Therefore, OMZ, while having ramifications for the euphotic zone carbon fixation, to some extent, OMZs seem to incentivize the CO2 drawdown via chemoautotrophic carbon fixation. This motivated us to experiment this, to measure the chemoautotrophic carbon fixation rates in one of the major oxygen minimum zone of the global ocean that lies in the Arabian Sea. Therefore, we sampled seawater in Arabian Sea across a biogeochemical gradient that ranges from northern to southern Arabian Sea. We sampled a total of 14 stations, where one stations belong to the coastal stations, and some of the stations in the northern Arabian Sea, and some of the stations in the southern Arabian Sea. We sampled seawater during winter monsoon, and during winter monsoon, the Arabian Sea faces convective mixing, which is indicated through the temperature and salinity profile of the sampling stations. We found that the sea surface temperature of stations from SK2 to SK7 was lower in comparison to the remaining stations. In contrast, the sea surface salinity was higher from SK2 to SK7 stations in comparison to the rest of the stations. This is an indication towards that convective mixing is happening towards 10 degree north of the Arabian Sea, and this has already been validated previously by previous studies that convective mixing largely occurs above 10 degree north. Therefore, we have categorized stations from SK2 to SK7 as convective stations, Except the coastal station, we have categorized the remaining stations as non-convective stations. The effect of this vertical mixing through convective mixing is indicated, can be shown in the nutrient profile. For example, the NOx concentration, which is the summation of nitrate plus nitrite, is below detection in rest of the stations, whereas it is above detection in the surface waters of the convective stations, meaning through convective mixing, the nutrient-rich subsurface water gets mixed with the nutrient-depleted surface water. Similarly, the phosphate concentration was higher in the surface waters of the convective stations. And the effect of this flux of nutrient from subsurface to surface waters is indicated through the chlorophyll concentration. For example, the chlorophyll concentration is quite higher in the convective stations in comparison to the rest of the stations. Arabian Sea also possesses oxygen minimum zone in its subsurface water, and Arabian Sea possesses the largest oxygen minimum zone. It ranges from around 150 meter to 1000 meter depth. This oxygen minimum zone provides a suitable niche for denitrifiers, where denitrifier reduces the concentration of nitrate, ultimately to nitrite concentration. And this nitrite concentration, which is a reaction substrate for Anammox bacteria, helps them to sustain their mechanism. We sample seawater depth from surface to around 1000 meter depth, where and we can see that the circles indicate the sampling depth. The open white circles represent the euphotic zone sampling depth. The solid black circles represent the sampling depth corresponding to the oxycline region, where oxygen concentration was greater than 60 but lower than 120 micromolar per kg. The solid white circles represent the oxygen concentration of suboxic waters, where oxygen concentration was lesser than 20 micromolar per kg. And the yellow solid circles represent the hypoxic water sampling depth, where oxygen concentration was greater than 20 but lower than 60 micromole per kg. We observed that dark carbon fixation rates were highest in the oxycline region, indicated through this purple color. We attributed this highest dark carbon fixation to ammonium oxidizers. Ammonium oxidizers are one of the most abundant organisms below the euphotic zone. And in one of the study in Eastern Tropical South Pacific, it has been observed that ammonium oxidizers could contribute about 50% of dark carbon fixation. We have called carbon fixation in the twilight zone as dark carbon fixation, meaning ammonium oxidizers has been reported to represent about 50% of dark carbon fixation occurring in the oxycline region. On average, the dark carbon fixation of suboxic waters, indicated through this blue color, is significantly higher than the dark carbon fixation in the hypoxic waters, indicated through this black color. And it is clearly visible in this figure as well. Now, the suboxic waters and hypoxic waters are different waters, and they provide different suitable stimulus for a different kind of organism. For example, suboxic waters are dominated by Anammox bacteria, autotrophic denitrifiers, nitrite oxidizers, ammonium oxidizers, methanogens, and sulfur oxidizers as well. Whereas hypoxic water is dominated by nitrite oxidizers, ammonium oxidizers, and aerobic methane oxidizers. Therefore, the abundance of these organisms, or the large abundance of these organisms in comparison to the hypoxic water, resulted in higher dark carbon fixation rates in the suboxic waters. We also tried to assess the contribution of Anammox bacteria towards total dark carbon fixation occurring in the suboxic waters, and we found that it could range from 1 to 47%. Meaning, while OMs, and it is clearly indicated that OMZs, which are reducing the concentration of nitrogenous nutrients in subsurface waters, to some extent, they are facilitating the CO2 drawdown by chemoautotrophic organisms. We also tried to assess the potential environmental controls that are playing a crucial role towards facilitating the dark carbon fixation. For example, we found dark carbon fixation was positively correlated. I think there's a mistake. Yeah, it was positively correlated with nitrite concentration. This nitrate concentration, which provides a reaction substrate for Anammox bacteria, indeed provides a suitable stimulus for Anammox bacteria. Therefore, the coupling among dark carbon fixation, nitrate concentration, and previously reported presence of Anammox bacteria in the oxygen medium zone of the Arabian Sea hints towards that Anammox bacteria are responsible for the dark carbon fixation in the suboxide waters. Additionally, we also found that dark carbon fixation was positively correlated with dissolved organic carbon concentrations, and it is already backed by an evidence by Zhao et al. 2017, where they have found that a significant increase in dark carbon fixation after the addition of glucose. We also found that dark carbon fixation, just like primary production, is positively correlated with particulate organic carbon concentration. This hints that dark carbon fixation too plays a crucial role towards CO2 drawdown and sustaining the food web by providing a new organic matter within the twilight zone. In this first figure, we have represented sea surface primary production, and it is clearly visible that sea surface primary production is quite higher or significantly higher in the convection dominated regions in comparison to the non-convective stations, which is facilitated by the flux of subsurface nutrient rich water to the nutrient depleted surface waters. Similarly, similar trend was observed in the euphotic zone integrated primary production. But the twilight zone integrated carbon fixation rates, meaning dark carbon fixation rates within the twilight zone, were more or less similar, or they did not show any such kind of trend between the northern and southern Arabian Sea. Therefore, we extrapolated the dark carbon fixation rates to the total global ocean, and we found that they could contribute up to 7.4 petagram carbon per year, which amounts to about 15% of the global ocean primary production, which is around 50 petagram carbon per year. Therefore, we concluded that dark carbon fixation also fuels the carbon demand of heterotrophs, which previously has been suspected that or hypothesized that the demand of heterotrophs within the subsurface water is not completely satisfied by the organic matter that is transferred from the euphotic zone. Therefore, there is a possibility that dark carbon fixation sustaining the heterotrophic activity of twilight zone as well. Dark carbon fixation can also act as an alternative source to intensify the oxygen medium zone of the Arabian Sea. And this has already previously been hypothesized that the amount of organic matter is not enough to sustain this much amount of oxygen medium zone in the Arabian Sea. Additionally, we have found that dark carbon fixation contribution to sinking carbon fluxes in the organism, meaning there is a hypothesis by Langer et al. That dark carbon fixation plays a crucial role towards and contributes to sinking organic matter in the OMZ of the Arabian Sea. And since we have found dark carbon fixation rate, there is a possibility, or it is an evidence that dark carbon fixation really playing a crucial role towards sustaining the flux of organic matter to the OMZ of the Arabian Sea. To summarize, dark carbon fixation rates raised up to 7.5 petagram, which amounts to about 50% of the global ocean primary production. And they are also responsible for sustaining the heterotrophic demand of organic matter in the twilight zone. And to conclude, dark carbon fixation do act as an alternative source required to intensify the OMZ of the Arabian Sea. Thank you.
Great, thank you so much, Himanshu. That was fascinating. Can you hear me or see me?
Yeah.
Okay.
Should I stop sharing? Yeah.
Yep, great. All right. Yeah, again, fascinating work. Does anyone have any questions at this point? No? All right. Well, I have one, and it's quite out of scope, I'm sure, but it has to do with sort of the net effect of the chemoautotrophy on the carbon cycle. And you mentioned that, it could possibly play a big role in the carbon sink. And I just wondered if you considered, so I work on the geovertical migration of critters from the ocean twilight zone and how they bring down carbon. And I just wonder if, there's a food source down there that potentially they could bring it back up, and whether or not that's something that you've considered in this this net effect of dark carbon fixation on the carbon cycle.
Did you say like you work in crustaceans?
Crustaceans, fish, yeah.
But they are heterotrophic, right? Meaning they are not fixing carbon. They are in fact utilizing the already available organic matter that is present within their surrounding, right? Meaning they are not fixing CO2 into their organic matter. Rather, just like humans, they are utilizing already fixed organic carbon. For example, on land, photosynthesis is performed by plants. Okay, so they are fixing carbon dioxide, but we do not fix carbon dioxide. We in fact use the organic matter through plants. Similarly, fish and crustaceans, being heterotrophic, they are not dependent on CO2, but rather they are dependent on the organic matter derived from primary producers. Therefore, while calculating carbon fixation, so it represents only the autotrophic activity, but not the heterotrophic activity by crustaceans or fish or other zooplanktons.
Right.
I hope I answered your question.
Yeah, no, I guess it's just the idea of another food source down there for potential heterotrophic organisms. But again, that's probably quite out of scope, but something that is really interesting going forward. Let's see, I think, yeah, we have another question. So Yibin asked if you can explain a little bit about how you scale your regional DCR observations to the global estimate.
Okay, so we categorized our dark carbon fixation rates on the basis of oxygen concentrations. So we had suboxic waters where oxygen concentration was less than 20 micromole per kg. We had hypoxic waters where concentration of oxygen was greater than 20 but lesser than 60 micromole per kg. And we had also oxycline region where oxygen concentration was higher than 60 but lesser than I think 120 micromole per kg. So what we did, there are already global estimates of global ocean, the volume of water with such oxygen concentrations. So there is a fixed volume of water with suboxic waters, and similarly there is a fixed volume of water with hypoxic waters. So what we did, we extrapolated our carbon fixation rates from suboxic waters to the suboxic volume of the global ocean. Similarly, we did the same exercise for the hypoxic waters, and we did the same exercise for oxycline waters. So what we did after extrapolating these carbon fixation rates to the suboxic waters, hypoxic waters, and then oxycline waters, we added them, and we got to know the total volume, the total carbon fixation rates occurring in the global ocean, but they are restricted to those oxygen waters. Means the concentration of oxygen. Also, there is one thing to note that the carbon fixation rates were occurring in the whole twilight zone. So there is a since we measured up to 1,000 meter depth only, there is a possibility. In fact, there are observations that carbon fixation is occurring in up to 4,000 meter also. And ammonium oxidizers, being the abundant organisms, they are present everywhere below the euphotic zone. So whatever the carbon fixation is occurring, ammonium oxidizers might be playing a role. Therefore, we could extrapolate it to the global ocean with considering the previous observations and the present exercise.
Great, thank you. Let's see, does anyone else have any questions for Himanshu? No? All right, well.
Thanks.
Well, yeah, yes, definitely. Congrats on your publication.
Thanks a lot.
Yeah. All right. Well, thank you so much, Himanshu. That was really fascinating, and thank you for joining us. And I